First, let's take a look and see what tonicity does with animal cells, right? Remember that one of the unique things that animal cells don't have is actually a cell wall, right? This protective, rigid outer coating. And so when you take a cell like an animal cell that doesn't have a cell wall, it's going to be a little more highly susceptible to changes in tonicity. So our example here is the red blood cell. And you guys are actually going to get a chance to look at red blood cells in a variety of different tonicities in lab once we get to Unit 7. So first of all, a red blood cell. Red blood cell is an animal cell, doesn't have a cell wall. And in the middle here, you can see that the red blood cell usually has this concave sort of disc shape to it. And this is the normal state for a red blood cell. This is the way that red blood cells like to hang out. And so red blood cells prefer to hang out in an isotonic solution. This means that the solute concentration on the inside of the cell is going to be exactly the same as the solute concentration on the outside of the cell, right? So in this case, water still moves in and out of the cell. It does so at the same rate. So the cell neither gains nor loses water. This is normal for animal cells. If we were to take now one of these red blood cells and instead put it into a hypotonic solution, this means that there are far more solutes on the inside of the cell than the outside, right? So in this case, water is going to chase that high solute concentration on the inside of the cell. Water is going to rush into the cell, and the cell is going to get bigger and bigger and bigger, and there's going to be nothing to stop that cell from overfilling. If it overfills, it pops. And this popping or this bursting process we call lysis, right? So we would say that the cell has lysed. And I highly encourage you guys to practice some of these technical terms that we use to describe what happens to these cells in these various solutions with various tonicities. All right, let's do the opposite. Now let's take the red blood cell and let's place it into a hypertonic solution. This means that the red blood cell is going to be exposed to a solution that has higher solutes on the outside of the cell than the inside. Right, so if this is the case, again, water chases high solute concentration. Water is going to flow from the inside of the cell now out. So the cell is going to lose water and it's going to shrivel. Now the proper term for this shriveling process for the red blood cells is a process that's called actually crenation. So when this red blood cell shrivels, and we want to spell the proper term for that shriveling process, it's spelled crenation, or we can say that the cell has crenated, right? So that's going to be your technical term for the shriveling of the red blood cells when you place them into a hypertonic solution. Under the microscope, the cells are going to look like little popcorn underneath your cover slip. Okay, so that's what happens to animal cells that are lacking a cell wall. Now, there are plenty of other organisms also that don't have a cell wall, and they have mechanisms which help them to combat taking up this excessive amount of water when they're existing in a solution that has far less solutes than the inside of the cell. And this process in which cells can control either solute concentrations or water balance is called osmoregulation. And this is really important for things like freshwater protists. So protists that live in ponds, for example, like the paramecium that you see in the picture below. This guy doesn't have a cell wall, nothing to protect it from an excessive uptake of water. And it's living in essentially a hypotonic environment, which means that the paramecium itself is hypertonic to the environment. So if the paramecium itself has more solutes than the outside world, the water from the outside is going to flow into the paramecium and it's going to continue just expanding and expanding and expanding. Right? If this were a red blood cell, it would lyse. But in the case of the paramecium, it actually takes that excess water that comes into the cell and it collects it within that contractile vacuole, right? One of those types of vacuoles that we talked about in chapter 6. And once the vacuole gets full, then it has the ability to contract and it pushes all that water back out of the cell and it prevents the paramecium essentially from popping and bursting and then dying. right? So it's a way that it has adapted to living its life within a hypotonic solution. Now for plant cells, it's a little bit different. Plant cells, remember, have a cell wall, and the cell walls actually help to maintain water balance in the plant cells so they don't end up popping. So let's take a look at what happens to plant cells as you place them into a variety of different solutions. So the normal condition for a plant cell is actually to be placed within a hypotonic solution, right? So this first image right here, in this case, this is what we would call normal for a plant cell. When you put a plant cell in a hypotonic solution, that means there are more solutes on the inside of the cell, water flows into the cell, and the cell expands, and it gets bigger and bigger and bigger, just like what we have for the animal cells. Except there is a limit to how big that plant cell can get because it has that rigid cell wall. And pretty soon that cell wall gets pretty full. The cell wall says, you know what, cell, we are done taking up water. This is as much as I can hold. You're not going to take up any more. And so water essentially stops flowing net into the inside of the cell, and the cell remains just full with a lot of pressure up against that cell wall, and it's happy this way. And this condition is what we call turgid. So that would be the technical term. It's written down here, the technical term for that happy, really, really full plant cell. Now, on the other hand, where an animal cell would be happy is here in the isotonic solution. The plant cells, not so much. These guys actually become very limp, or what we call flaccid, within the isotonic solution. Yes, water comes into the cell. Yes, water leaves the cell, but it does so at the same rate, and the cell doesn't swell and it doesn't get full. It doesn't exert that pressure up against the cell wall, and this is where your plants are going to be kind of droopy, and they're going to look unhappy. And then the last condition is where you put a plant cell into a hypertonic solution. And again, you guys are going to get a chance to see this, hopefully, in lab in the osmosis and diffusion unit. And if you take a plant cell and you place it into a hypertonic solution, it's going to lose water, right? There are more solutes on the outside of the cell, so all the water from the inside of the cell is going to flow out. And while the cell wall doesn't move, right, because it's pretty rigid here on the outside, the plasma membrane, right, is actually going to end up pulling away from the cell wall, as you can see here, right? And that's going to allow the inside of the cell to sort of shrink as the cell loses that water. And this process is called plasmolysis, or we say that the plant cell has plasmolized. And this is usually a death sentence for the cell. The plant cell is not going to come back from a plasmolized state. And so just to give you a nice little preview of what that's going to look like under the microscope, these cells should look familiar. These are your Elodea cells, and they're going through the process of plasmolysis. You can see here the plasma membrane has pulled away from the cell wall here of this cell, and all of those little chloroplasts are getting bunched together in the very center there. Okay, so far, what we've talked about is the diffusion of components across a selectively permeable membrane, completely on their own. Now, remember at the very beginning of this chapter, we talked about how there are certain substances that can move across the membrane on their own perfectly fine. These are going to be anything that's either hydrophobic or nonpolar and very small and can easily pass across the membrane unaided. There are other substances that need a little bit of help, though. They still want to pass across that membrane down their concentration gradient, but maybe they're polar molecules or they're charged ions, and they're afraid of going through that hydrophobic core. And this is where the cell will turn to a slightly different kind of diffusion. It's still diffusion, but it's what we call facilitated diffusion, because these molecules or ions need a little bit of facilitation or a little bit of help to get across that membrane. And the cell is going to actually end up using transport proteins to help get these particular molecules or ions across the membrane down their concentration gradient. So let's take a look at the proteins that are involved in this process of facilitated diffusion. First of all, we have the channel proteins, and second, we have the carrier proteins. So these transport proteins, again, are going to allow the passage of any hydrophilic or polar substances across the membrane. How channel and carrier proteins differ. We're going to take a look at each on the next few slides individually, but just a preview. Channel proteins are going to essentially have a hydrophilic channel that certain molecules or ions can use kind of like a tunnel, a safe passageway to get to the other side. A good example of a channel protein we've already talked about is the aquaporin on the very first slide of this chapter, right? So the aquaporin is going to help or facilitate water as it passes across the membrane. The other kind of transport protein are carrier proteins. These guys actually bind to the molecules and they change shape a little bit as they pass those molecules across the membrane. So they kind of hug the substance that they're shuttling across, whereas the channel proteins are just essentially a tube or a channel for the substances to get across. One of the most important parts, though, about these transport proteins is that both of them, channel and carrier proteins, are usually very specific to that substance that it is meant to move across. So, for example, aquaporin, the only thing aquaporins move across the membrane is water. They can't move any sugars, they can't move proteins, they can't move ions. They are just meant for water, and that's it. So each type of channel protein, each type of carrier protein will be specific for that substance it shuttles across. So let's take a look a little bit in more detail about these channel proteins, right? So down below is an example of a plasma membrane here and a big purple channel protein embedded in it. And so you can see that the channel protein is going to be one of these transmembrane proteins that spans all the way across the membrane, and it creates this nice little channel or tunnel for these molecules or solutes that otherwise couldn't pass across the membrane on their own. It gives them that safe passageway to, again, diffuse down their concentration gradient from an area of high concentration to an area of low concentration. Again, aquaporins are channel proteins that facilitate diffusion of water. There are also ion channels that facilitate the diffusion of ions, right? Ions are charged particles. They're not going to get across the membrane on their own, and yet the cell needs them and wants them to come into the cell or maybe out of the cell. And so it needs to produce these ion channels to allow various ions to come across the membrane. Where ion channels are unique on another level is that some of them are what we call gated channels. And these gated channels actually have the ability to open or close in response to a stimulus. So it's not just good enough to have that particular ion channel in there to allow, let's say, that ion to come across, but sometimes the channel is closed and sometimes it opens. It all is dependent on what the cell needs and whether that stimulus to open it or to close it is actually present. So it gives the cell that one extra layer of being able to regulate when it allows certain things to come across the membrane. All right, carrier proteins. These guys are going to hug that particular component that it's allowing to come across the membrane. So again, down here we have our plasma membrane, and a carrier protein will oftentimes be open naturally to one side of the membrane. When it's open to that side, it allows a solute or maybe multiple solutes to come in and dock to a certain part of that carrier protein. Once that solute docks there, it actually stimulates that carrier protein to change shape. When it changes shape, it flips open to the opposite side of the membrane, and then it can release the solute on the other side. So again, this is still going to be diffusion, either through the use of a channel protein or a carrier protein, so the solutes are still moving down their concentration gradient, and the cell doesn't need to add any of its own energy to perform these functions. All the proteins are doing is giving those molecules or ions that otherwise wouldn't be able to pass across the membrane the ability to actually do so.